Hello, everyone. Here we're going to be talking about the blood-brain barrier, which is like a security system for the brain. So, what is the blood-brain barrier? Well, it's going to be this special layer of cells that lines the blood vessels in the brain, and it will control what can pass through from the blood into the brain. And this barrier actually keeps out harmful substances like bacteria, for example, and toxins, while allowing important things like nutrients and oxygen to be able to get through. And it helps protect the brain and keep its environment stable as well, which is going to be crucial for its proper functioning, as you can imagine. In fact, with this information, we can add that the blood-brain barrier has both structural and functional components. What are the structural components? Well, the structural components of the blood-brain barrier consist of tight junctions that seal together the endothelial cells of the brain and capillaries, like we can see over here, and the processes of many astrocytes, as seen on this image as well. And astrocytes are a type of glial cells that will help to maintain the nervous tissue. Therefore, notice how the extensions of the astrocytes, they're actually pressing against the capillaries, and they will also secrete chemicals that maintain the tightness of the tight junctions. And because an endothelial cell plasma membrane, like any other plasma membrane, has a hydrophobic interior, lipid-soluble substances are going to be able to diffuse across the walls of these brain capillaries, whereas most water-soluble substances are going to be unable to do so. In addition, we can say that since there are no pores or spaces between the tightly sealed endothelial cells, there's going to be large substances such as cells and proteins, for example, that are also not going to be able to move across the walls of these brain capillaries. This means that we can say that the structural component of the blood-brain barrier allows the fusion of lipid-soluble substances across the brain capillary walls, but it will prevent the movement of water-soluble substances and cells and proteins between the endothelial cells. Now, the functional components of the blood-brain barrier, they consist of membrane transport proteins, either carrier and ion channel, in the endothelial cell plasma membranes that will selectively move some water-soluble substances across the brain capillary walls. If a water-soluble substance does not have a membrane transport protein in the endothelial cells of the brain capillaries, then that substance will not be able to cross the blood-brain barrier. But of course, there will be exceptions to this rule, and the main exception is actually water. So although water is a polar molecule, it can diffuse across the endothelial cell plasma membrane without the use of a membrane transport protein. And why is this? Well, it's because water is actually a very small molecule and it's also uncharged, so it doesn't have a charge to it. Now, within the choroid plexus, these endothelial cells, they actually have smaller gaps, and this allows a little bit more of large particles to be able to cross over. Let's talk now about the importance of the cerebrospinal fluid, which is going to be this clear, colorless liquid that protects the brain and also the spinal cord from either chemical or physical injuries. And it also carries oxygen, glucose, and other needed chemicals from the blood to the neurons and the neuroglia, or the glial cells. So CSF is going to be continuously circulating around the brain and the spinal cord, not only in the subarachnoid space, but also in the central canal of the spinal cord and through the cavities in the brain that are called ventricles. Now, CSF is actually produced by the choroid plexus, which is present in every ventricle. We can actually see them present here in the lateral ventricle, in the third ventricle, and in the fourth ventricle as well. Now, the volume of CSF is around 80 to 150 mLs, and this corresponds to about 3 to 5 ounces in an adult. So CSF will contain glucose, it will contain proteins, it will contain lactic acid, it will contain urea, cations like sodium, potassium, calcium, magnesium, and anions such as chloride and bicarbonate. And it also contains some leukocytes. And because it contains all these types of substances, we can say that CSF contributes to homeostasis in three main ways, which are mechanical protection, chemical protection, and circulation. When referring to the mechanical protection, CSF will actually act as a cushion, absorbing the shocks that might otherwise injure the brain and the spinal cord, as these are very delicate tissues, and it also helps the brain float inside of the skull. Then we have chemical protection, so CSF will keep the chemical environment stable, and this is for proper brain function, so even small changes in its composition can affect the brain activity. So it's important to maintain the balance always. With regards to circulation, well, CSF will help in exchanging nutrients and waste between the blood and the brain. Therefore, we can see that CSF has several very important functions. And we already know, like I said, that CSF flows from the ventricles into the spinal cord and then the subarachnoid space. And it is slowly absorbed back into the blood through these small projections that are called arachnoid villi. We can see them right over here within the superior sagittal sinus. We can actually see these arachnoid villi on this image as well. There's one over here and one right over here within the superior sagittal sinus. Now the body produces and reabsorbs CSF at about the same rate, so about 480 mLs per day. And this means that the entire volume is going to be replaced around 3 times per day. And this balance actually keeps the pressure of the CSF constant, usually around 10 millimeters of mercury. The cerebrospinal fluid is made in the choroid plexus. I did mention the choroid plexus in the previous slide, and I said that they are located within the brain ventricles. But what is the choroid plexus? Well, choroid actually means blood, and plexus means group. Therefore, the choroid plexus is a network of blood vessels in the brain ventricles. And this means that we will find the choroid plexus in the lateral, third, and fourth ventricles, like we saw on the previous slide. And the capillaries present within the choroid plexus are covered by ependymal cells, which are these cells lined up right over here. And the function of these ependymal cells is to filter and secrete CSF from the blood plasma. And notice how these cells are also tightly joined, and they form this barrier that controls what can enter into the brain tissue. And of course, this will protect the brain and the spinal cord from any harmful substance. In these models that we find in the lab, we can see the choroid plexus present in the ventricles. However, on these models, we cannot see the choroid plexus within the lateral ventricles. But we can see over here the presence of the choroid plexus in the third ventricle, represented by this pink line on top of the third ventricle. We can see it over here on this model as well, which is being represented by this gray line. And if you look closely, you will see that this gray line has actually several small red and blue lines as well. And this is basically representing the blood vessels within the choroid plexus. And we can see the same thing on this other model, which actually is a little bit better to see. So notice how there are these wiggly lines that are red and blue representing the arteries and the veins within the choroid plexus. As you can imagine, the brain needs this constant supply of oxygen and glucose from the blood to be able to function properly. And although the brain is only 2% of the body's weight, it actually uses about 20% of the oxygen and glucose even when at rest. So neurons will rely on glucose and oxygen to be able to produce energy. And when a part of the brain becomes more active, this means that it receives more blood flow. And if blood flow to the brain slows down, it can cause unconsciousness. Now, if there's a lack of oxygen for even 1 to 2 minutes, it will affect the brain's function. Now, if you stay more than 4 minutes, it can actually cause permanent damage. So since the brain doesn't store glucose, there needs to be this continuous supply of glucose within the brain. Now, if glucose levels drop, this means that the individual can even get confused, can feel dizzy, can even have seizures, or even faint. So there needs to always be this constant supply of glucose within the brain.